Now let's look at correlation engine. So on your same browser, click on base correlation engine. Actually, if you look over here on the right, if you're on the home page, you say correlation index contact index. There are 22,261 studies in their database, and this represents everything. Sequencing, genomic, proteomic, everything that they can get their hands on is in this database. Once they've downloaded all these studies, so all these studies have associated variables, right? Cancer, or they use this chemical. What they've done is they've already done the statistics for us. So basically, out of these 22,000 studies, there's actually millions and millions of gene lists that they have generated that we can now compare. We can actually browse through, or we can use our data compared to theirs. Here's what I want you to do is, in this network, I want you to click on curated studies here. It's at the top. Here's what I want you to do is, I want you to put in your favorite cancer, whatever your cancer you're working on. I'm going to put ovarian. And as you type, you'll see a phenotype probably come up. I'm going to click ovarian cancer. In this database, I can look through 300 studies for ovarian cancer. I didn't generate any of this data. That's millions and millions and millions and millions of dollars worth of research that I can get access to. I got 300. Anybody get more than that? What'd you get?
1,095.
What was it?
Breast cancer.
Breast cancer. Yeah, totally. Yeah, you picked the right cancer. You got 6? That's all right. 6 is better than 0, right? What you find, though, is a lot of times, I'll throw a gene list for lung cancer in here, and I'll get a bunch of bladder cancer studies back. Like, my cancer looks more like bladder cancer than it does lung cancer. Don't be fooled by these, phenotype, characterizations we put on cancer. Again, what's nice, too, is I can basically build these meta-analysis off of any cancer that I want. I can pick these graphs. If I look here, I can grab some of these lists and just analyze those as well. I can filter this any way I want. I can filter it, say I only want somatic mutations in human, in late stage cancer. I can totally get those samples. Okay. For our study, we are going to look at, so what I've done, so go to collaborations here. So click on collaborations. You should see MBCO AACR 2018 gene list. Join that list. And if it kicks you off, go back to collaborations and click on it again. What you should do is you should get to this panel here. Is everybody here?
Do we join the group?
Yes, join the group, please. So what I've done is I've taken that almost 2,800 genes that you guys analyzed in an RNA-seq study. I was only interested in things that were overexpressed in the macrophages. So basically what I did was I just did a log 2-fold change and then took all those genes. So we have 947 transcripts upregulated or overexpressed in the macrophages, at least 2-fold higher than what you'd find in the tumor. Does everybody understand that? All right, so click on MBCO.
I don't get the project name.
All right, you should see a list that says ovarian cancer versus tumor-associated macrophages. Click on that. And then another list will come up. It's like those Russian dolls. Click on this text. So at OC versus TAM 7x7. Okay, given here on this first is basically all the genes in the gene list that I downloaded, along with its associated fold change, and then there's p-value information. Okay, so let's look at some of these. So that's my goal is given a list of 2,800 genes, or even, in this case, 940 or 841, I don't want to I want to use the information. I want to like try to use all of it at the same time. I think a lot of people, this is what kills me, is I'll do an experiment for somebody, they just done like an RNA-seq or just global transcription. I'll get the results, and then the first thing they ask me is like, what did my gene do? Like, what did my gene do? Or what did my pathway do? It's like, well, you got all the data. Like, why would you focus just on that? You know, why don't you let me show you like everything, and then you can maybe pick and choose. So what this is allowing you to do is get to the story. So let's click on pathway enrichment. So it's just like string and all the other David and all the other programs that you have out there. It's going to do the same thing, but it allows you to filter it a little better. So if I look up here, again, we're seeing that innate immune response, right? Click on this. So what this is telling me, the innate immune response, here's my gene list. I put 841 genes. They're all going up. There are 479 total genes known to be associated with the innate immune response. Doing a Venn diagram, I get 88 genes in common. The odds of that happening are 7 times 10 to the negative 59th, right? We did kind of enrich for that, and if you wanted to basically grab those genes, they're all here, and you can actually export that. What's also cool is now what I could do is I could say, okay, I'm really interested in this innate immunity. I'm going to download this list, and then I could throw it back into string and see what it does, right? Bioinformatics is going from small to big to small to smaller to really big. You can go any direction you want. It's not just one direction. Okay. So we're here, innate immune response, and again, this is giving you just kind of the information here. You can see hemostasis. I've been kind of looking at this data set. It's unique. In the paper, they say that they believe these macrophages are stimulating angiogenesis, and I actually can see evidence of that in the transcriptome. Okay, here's what we can also do. Again, we can look for many different things. This is just giving you all the possible categories. What we can also do is look at regulatory motifs. So click on that and hit apply filter. Now what we're doing is we're looking for overrepresented transcription factor binding sites in the genes that we've just isolated. Again, this gives you a way to get to that upper level. A lot of transcription factors don't change at the expression level. Again, they're getting phosphorylated, they're getting, translocated to the nucleus. You don't see the change in transcription. But I'm willing to bet that ELF1 is probably playing a role in my system in those macrophages, even though I probably don't measure it at all. And not only that, it's affecting all these genes here, right? That's a big effect. Why would I pick something below that? If I really want to affect this system, I'm going up a level, right? And that's what I'm using this software for, is to not only understand my system, but to find the inflection points where I can get at it. Okay. How are we doing on time? Not too bad. Okay. Let's go to, and then another way to get to that upper level is we can go to Knockdown Atlas. So click on that. It's at the top. This I really like. Obviously, we've knocked out just about every mouse gene there is, right? We have a record of this. All of this knockdown information is in the database. When you knock a gene out, it affects certain genes. Again, it makes that wave in the transcriptome. What I can do now is, based on the genes that I put in, what it's telling me is when I knock this thing out, interleukin, or what is this? I know, I get that wrong every day. You'd think I'd get it right on the last day, but no. So you knock it out. When you knock this gene out in this study, our genes go up. This gene is probably playing an inhibitory role in the system that we're analyzing, at least in the macrophages. We can look, and there's some that are negative. you can go to the actual study and look at that information. So I clicked on, so on perturbed genes, I just clicked on this, it kicked me out to another window. Basically, this is showing me, here are all the genes that changed in their system, here's the ones that changed in mine. There's 322 that are common, and 313 of them went up in both studies. The odds of that happening by random chance are 6 times 10 to the negative 160th. After a while, the p-values just don't even matter. You're just like, okay, whatever. I actually talked to Illumina. I debated. I didn't think they could calculate p-values that small, and they've basically spent an hour convincing me they can. So they got some really high-duty, computers. Okay. So again, another way to find things that are related to your system that you're not measuring directly. And again, I would argue that these are probably your important, main players in your system. Again, we could take this, we could look for gene knockouts, we could look for things with mutations, overexpression. If I overexpress a particular gene, does it have any effect on the genes that I put in? Lots of ways to cut this cake. And the greatest thing is, I am not trusting what somebody says about their data. I'm actually mining the data itself. Does everybody understand that? That whole speech, Bill with the, reproducibility in academia, it's true. That there's probably 100,000 studies in gene expression omnibus. This is where we're sticking all our data. Illumina went through it and found only about 25% that they can use, right? It's a huge deal. So again, you can say whatever you want about your data, but you can't fake 30,000 different things. And if you can, then you probably shouldn't be doing this. You should probably be doing something else, right? I love this because I'm looking at their data and I know exactly, what mine's related to without having to trust what, somebody trying to get a grant saying. Okay, let's go up. You're gonna like this. This is the drugs, Pharmaco Atlas. Just like with the Knockdown Atlas, a lot of these databases have access to, in all of these studies, people tell you what they've added to their studies, and there's a lot of drugs being used. When the drug's been used in an experiment, we know what genes are affected in that one. So we can actually go here and we look at what genes either have a positive or negative effect on our genes. If you look at this first one, pristane. Has anybody heard of pristane? Either did I. But I looked it up. And pristane is used to induce autoimmunity in rodents, right? We saw the innate immune response in the other one. We saw it in string. This is kind of how I analyze. And there's, like I said, it's not a process. It's more, kind of viewing the data, different sections of it. Is there a theme that kind of develops? And I'm getting this innate immunity. And if you actually went down, there's also this whole angiogenesis thing going on. Here's what's cool about Pharmaco Atlas is I can say, hey, I got a list of genes that say in my patient that change, that are different than their normal tissue. Instead of maybe targeting one of those genes, I put their whole profile in here and I find a drug that affects most of those genes. Maybe I find a couple of drugs, right? Again, what this allows you to do is see the story before you actually do the experiment. And it's going to allow you to do better experiments because it's kind of like getting the cliff notes before you even go in there, right? I should expect this. I might not know exactly the details, but given all this transcriptome data, I would expect this. So I'm going to inhibit this, right? Okay, so that's Pharmaco. And again, you can click on any of these things and look them up, look at the studies, download all those genes that are common. Let's go to Disease Atlas. Okay, so we put our gene list in. Again, this is a very macrophage-centric gene list. Not only that, but probably associated with different types of cancer. We can look and see, based on what we put in, what other systems are probably, pissed off macrophages playing a role? If we look, skin disorders have a positive correlation to our gene list, right? Contact dermatitis, you can imagine those macrophages infiltrating the tissue compared to normal. That also brings up other issues, right? If I'm going to use this as a macrophage signature, and there's a wound, a fresh wound by the tumor, maybe some of that macrophage signal is coming from that, and maybe not my tumor itself. Keep going down, you'll notice a lot of viral stuff, inflammatory. Keep going, you'll see an orange, it'll be cancer, I think it's right after heart and vascular disease. Now what this is doing is grouping it based on disease categories. And so these are all the cancers that are similar to our list. You can hit open 29 more to see the entire list. A lot of these tumors, or a lot of these types of cancers, probably don't have that as much macrophage invasion. Look at lung cancer. It actually has a negative correlation. I would argue there's probably more macrophages in normal, healthy lung tissue than the cancerous tissue. And you can see the types of tissue, and I would say the reverse is probably true with the kidney. Go down to ovarian cancer. So it's after malignant tumor of muscle. Everybody see it? Click on that. These are all the ovarian cancer studies in this database that are correlated to our gene list. If we look at that first one, it's basically what we did, except they actually have a better comparison, right? They took the inflamed dendritic cell and the normal dendritic and compared those two together. We're getting the same genes. The odds of that happening are about 8 times 10 to the negative 90th. 89th. Again, we can go and we can look at all those genes and download anything that's common. We can see what's different, what's common. What I love about this is there's so much data out there that people could stop running clinical trials, killing mice, growing cells, and I'd have enough data for 3 careers, easily, right? Millions and millions of dollars worth of research in here that you can leverage for your project. You don't even need your own data. And in fact, most of what I do is not analyzing other people's data. It's going in there and getting a bunch of data for them, right? Saying, hey, I know your study's great and everything, but it's only one study on, kidney cancer. I can go get you 10 studies and basically show you everything, basically combine all those and get you, an answer better than any one individual study itself. Is everybody familiar with Nate Silver? 538? You don't see very many statisticians that are famous, so anytime we get one of our brood that becomes famous, we're like, have anybody heard of Nate Silver? So we're all proud of him, by the way, and we all take credit for him. But the thing is, the reason he is famous is in 2012, he predicted just about every major election in this country, right? Do you think he asked anybody who they were voting for? No, he didn't. All he did was he took all the polls that were out there, right? Some obviously were biased left, some were obviously biased right, and when he took the average of all those polls, it was pretty much the exact answer. We can do the same thing with gene expression, I swear. And I have videos where I do meta-analysis, and it's gorgeous. So, again, I want to stress the fact that you can use this to analyze your stuff, or you can use it to go get a bunch of other stuff. And
What if you have the gene equivalent of Donald Trump?
What's that?
What if you have the gene equivalent of Donald Trump?
Gene equivalent of Donald Trump?
Because he said Hillary's going to win.
Nate didn't do so well in 2016.
He said Hillary's going to win.
Yeah, Nate didn't do so well in 2016.
Yeah, he did it. But actually, he was more accurate than most of the other. There's all kinds of bias and stuff like that, and surveying. Yeah, that's a good point for sure. Well, he was good in 2012. I think if you actually did some, I'm fascinated not only with elections, but I actually Bob and I used to be in a football pool, and we were really intense about it. His dad always won, though. But where was I going with this story? I don't know. What's that?
Sort of the red hair, the unexplainable, unexpected.
Yeah, you just never know. That's what I like about it is I don't like just one tool. I like lots of tools. And if 2 different tools are telling me the same thing, that makes me believe it a hell of a lot more, for sure. And the thing is with Illumina, I trust that they've processed the data correctly, and that's a huge thing. About the only bioinformatician I trust is this guy, and maybe Zoo a little bit. Just a little bit. Okay.
Mike,
Yeah.
are people using this as preliminary data, or can you like reasonably
Yes.
use this as preliminary data in your grant? Or
Yes. You can download other people's data and publish on it. It's completely legal. You won't get arrested. The statistical police will not arrest you. And in fact, you should. And that's the thing is just because somebody's published on their data, it's like an iceberg. You know, they mention about this much of their data, and there's all this huge, gorgeous, data down below that's what I love. Go ahead and publish on it. A lot of times I'll analyze their data and go, you were completely wrong. You totally missed this, So, I mean, there's a lot of data out there. There's a lot of gold in them there hills, and all you got to do is mine it. It's out there. Okay. Here's what I really like too is you can use this information. Once you've got a profile, now you can use it to find all kinds of stuff, right? Go to Body Atlas. This will tell you based on the genes that we put in and the direction that they're going, what they most look like. And it probably is not too big of a stretch of the imagination to know that these are immune cells, right? Here is what's really cool. Go to cell lines. Now I can find a cell line that kind of replicates whatever kind of profile that I put in. Imagine doing this with one of your patients, right? I've got a patient, I know what genes are different in the cancer. I throw those genes through this. What it'll do is it can maybe find me a cell line that most resembles my patient, right? Then I don't have to experiment on them. Maybe I take that cell line and start putting a bunch of chemicals on it. Yeah?
Does this just do a two-way comparison, or can you do more? Because we're just comparing two groups, right? There's just differential expression of one group over another group.
Right.
But if you had a bunch of different cell types and they all had different differentially expressed genes relative to others, would this be able to handle that and tease it out? Or?
What you would do is you would get it would be related to say you had a combination of cell types. It would basically tell you these genes are probably coming from your immune system. This is probably stromal. What you'll get is basically it'll be red or green, and you'll see things that are the actual opposite would be green. So you can kind of, based on this readout, you could kind of put together what your potential cell types are in the tissue that you're looking at.
Got it.
Yes.
Okay.
Yeah.
Can you compare between species? So if you have, if you're studying human cancer, we always try to study studying things in mice, but some of the mouse tumor lines may be better approximating the human.
Totally.
Can you do that?
Yep. Totally can.
On this?
Yes. There's a meta-analysis. What you can do is you can compare lots of things. So all of these gene lists are available, and what you do is, if you see something here oh, this is a cool study, I want this gene list. Actually, I don't think it's going to give me a gene list. Damn it. If I go to curated studies, now what I'm doing is I'm finding things that are most common to my gene list. And again, of course, macrophage stuff comes up, right? What you can now do is you can say, I want to compare these lists, so I can actually just grab these. So I'm going to get all of these genes that are different, get as many studies as I want, and then at the very end, what I do is go to this meta-analysis. It's got all the genes, or my studies. Now I can see what all the genes do in those. Right? And what's really cool is, I just kind of picked them haphazardly. But what you do is you pick, like, say I was working on papillary renal cell carcinoma, and I had like 8 different studies and 17 different lists in those studies, and they'd all like be green or red. I mean, the consistency is amazing. It's just like Nate Silver, right? It's just, you take the average, everybody's kind of seeing the same thing, maybe not in the exact, magnitude, but when you put all that information together, it's just incredibly powerful. Okay. Let's go to, so that is Correlation Engine. I think there's a lot of programs that are kind of replicating what this is, and you could probably find different programs that do different tabs, but I think this thing kind of brings it all together, and what I like about it is you can do interactive help. So you can actually click on that, and as you're going, you will get help. It'll tell you what things mean and what things don't mean. It's very, I mean, I'm a hippie at heart. I wish things were free. But the problem is the free stuff that I could show you, you probably wouldn't be able to use it very well, right? This, very easy to use, very intuitive, and I think you got enough time, like if you really want to go with this, you totally can, right? You can figure out all kinds of things in a couple months. All right, actually I think it's 5 months, but. Okay, so here's now what I want to do is, now what we're going to do is get to the, let's look at some of the clinical data. So go to Cohort Analyzer.